Thank you very much. I'm going to make some forward-looking statements during this presentation. The risks are summarized on this slide and on our website, which houses our SEC filings. Clearside is developing and delivering treatments that restore and preserve vision for people with serious chorioretinal diseases. Our versatile therapeutic platform includes our SCS microinjector with proprietary drug formulations that target the suprachoroidal space. We have both an internal R&D development pipeline as well as external partnerships that drive our pipeline further. The core advantages of treating via the suprachoroidal space include the ability to target chorioretinal tissues for potential efficacy benefits, the ability to compartmentalize drug away from the front of the eye for potential safety benefits, and bioavailability. We're essentially bathing chorioretinal tissues with drug. And for small molecule suspensions, we have prolonged drug levels for durability. Our pipeline is shown here. Our internal pipeline includes CLS-AX, which is a proprietary suspension of axitinib. It's currently in a phase 1/2 trial for neovascular AMD. We have external collaborations, one with REGENXBIO, who's using our technology to deliver their gene therapy, currently in phase 2 trials for wet AMD and diabetic retinopathy. We also have a partnership with Aura Biosciences using our technology to deliver their virus-like drug conjugates for the treatment of choroidal melanoma, currently in a phase 2 trial. They've recently announced that they're going to go to phase 3 with our technology as well. And then, as many of you know, XIPERE was approved last year for uveitic macular edema. Bausch + Lomb is commercializing that in the US and Canada, and Arctic Vision is commercializing that in Greater China and much of the Pacific. Next, I'm going to talk about CLS-AX, our proprietary suspension of axitinib for suprachoroidal injection. In this program, we're essentially marrying a very highly potent pan-VEGF tyrosine kinase inhibitor with the potential benefits of suprachoroidal delivery. This is some data that we've published from our preclinical studies. On the left, we show that CLS-AX injected suprachoroidally provides targeted delivery relative to intravitreal injection at the same dose. In fact, there's 11 times the amount in the posterior tissues when we inject suprachoroidally versus intravitreally. And then on the right, we show that CLS-AX has potential for meaningful durability out to 6 months. We can see levels at 1 month and 6 months. We can see a nice compartmentalization effect. In dark blue is the suprachoroidal space, and light blue is the retina, and in orange is the vitreous. And importantly, at day 182 or 6 months, these retinal levels are several log orders higher than the IC50 for the VEGF receptor 2. Next, I'm going to show some result snapshots from our 3-month final data, as well as some interim 6-month follow-up. OASIS was a phase 1/2 clinical trial in treatment-experienced wet AMD patients with active disease at screening. There were 4 dose-escalating cohorts. Patients were treated with a standard of care, 2 milligrams of aflibercept at screening. They're seen a month later at baseline, received CLS-AX, then they're followed for 1, 2, and 3 months with additional 3 months of follow-up, including the extension study. They undergo monthly assessment for additional treatment with aflibercept with protocol criteria. We enrolled heavily treated anti-VEGF patients in this wet AMD study. All of these patients were sub-responders with active disease at screening confirmed by an independent reading center. And we felt that enrolling these difficult-to-treat anti-VEGF sub-responders allowed observation of possible signs of biologic effect while minimizing any false signals. We found a very positive safety profile in all 4 cohorts, both in the 3-month final data and the 6-month interim data. Basically, there were no dose-limiting toxicities and no adverse events related to inflammation, vasculitis, or vascular occlusion. These are tables showing that CLS-AX reduced treatment burden across all cohorts. Here we show the average monthly injections before CLS-AX and the average monthly injections after CLS-AX. On the left are the reduction in treatment burden using all the additional treatment, and on the right is treatments per protocol criteria. And basically, there was a 73 to 100% reduction in treatment burden in cohorts 3 and 4. These graphs show the supplemental anti-VEGF injection-free rate up to each visit in cohorts 3 and 4. On the left, again, are all the additional therapies, and on the right are all the additional therapies per protocol criteria. And basically what we see is 75% of the patients had no additional therapy to month 6, and 88% had no additional therapy to month 5. This is a similar graph to similar table to what I showed you earlier. This is the 6-month interim data, and this shows the reduction in treatment burden on the left, all the additional therapies, and on the right, all the additional therapies per protocol criteria. Basically a 90 to 100% reduction in treatment burden in cohorts 3 and 4. These are graphs of the mean best corrected visual acuity letter score change from screening over time. You can see cohort 3 is in green, cohort 4 is in blue, the average is in black. And we see that the visual acuity is quite stable. Because this is interim data with declining sample size over time, there's more variability as we go on. But you can see even at month 6, the standard error bars cross the 0, no change line. On the left is all the data plotted, and on the right excludes data after additional treatment to be sure those additional treatments weren't driving these results. And you can see the curves are essentially identical. This is data plotted similarly, but here we're plotting mean central subfield thickness change from screening. Again, as time goes on, there's fewer subjects and there's more variability, but you can see in the overall average that the central subfield thickness is quite stable. On the left is all the data, and on the right is data that's excluded after additional rescue. Finally, I want to take you through one case. This is a 6-month case study where CLS-AX demonstrated a biologic effect in the anti-VEGF subresponder. This patient received 89 prior injections. At screening, in the upper left, the fovea is marked, the subfoveal fluid is shown, and a fibrovascular PED is shown. Visual acuity is 75 letters. They receive aflibercept at this visit. They come back a month later at baseline. You can see that the subfoveal fluid persists, the visual acuity is stable. They receive CLS-AX suprachoroidally. Then they're seen at month 1, month 2, month 3 in the bottom left. You can see that the subfoveal fluid's improving. Month 4, which is 1 over, the subfoveal fluid's completely resolved. In fact, 4 months after CLS-AX, the subfoveal fluid is better than 1 month after aflibercept. Then they're seen at month 5, and at month 6, the fluid comes back and they receive their first additional therapy with aflibercept. So in summary, CLS-AX showed promising safety results, durability, and biologic effect. Our next steps are to follow the remaining patients in the extension study, and we'll share that data in the first quarter of next year, and then we plan to start a phase 2 trial in the first quarter of next year as well. Thank you very much.